Hey, good morning. I think it's 9. We want to get started. Thank you for coming for this morning's MCC seminar. For those who don't know me, my name is Haibin Ran, and I'm associate professor in the Department of Integrative Biology and Physiology. So today is my great pleasure to introduce our speaker, Dr. Gensheng Feng from UCSD. I just want to give you a brief introduction about Dr. Feng. He got his BS in biology from Hangzhou University, China, and his PhD in molecular biology from Indiana University, Bloomington. That's in 1990. And he then did his postdoc training with Dr. Tony Paulson at Mount Sinai Hospital, Toronto. After his postdoc training, he comes back to Indiana University again, now at that time at School of Medicine as assistant professor, established his independent lab. He then moved to Brigham Institute La Jolla in 2000 as associate professor, and then in 2009 moved to his current institute, UC San Diego, now as a professor and in the Department of Pathology at School of Medicine, UCSD. Dr. Feng, his research has been really focused on oncogenes, and he was the first one actually identify SHP2, a tyrosine phosphatase. His lab also identified signal molecules, GRAP and GAP, and studied their functions in not in just the metabolism, but also mostly in liver cancer. Currently, his lab has been focused on deciphering signal crosstalks, between metabolism and cancer, and mostly studied their dual functions of these oncogenes as in tumor growth and also immunotherapy. Dr. Feng is editorial board members of multiple journals such as MCB, JBC, Journal of Hepatology, and Molecular Cancer Research. He's an associate editor of the journal Liver Research. In 2005, he was elected as a fellow of AAAS. Dr. Feng's research has been constantly funded by NIH since 1994. He currently holds 3 R1s and one P1 project and many other foundation grants and COI grants. Just one last note is that he has been really active in scientific communication. He has been constantly attending and also co-organizing many conferences. For example, last year, the FSAP Liver Biology Conference and also Colorado Spring Harbor Asian Conference on Liver Development, Metabolism Disorder, and Cancer. Oh, that's a long name. Without further ado, Dr. Feng, the stage is yours.
Okay, great. Yeah. Okay, yeah. Thank you, Haibin, for the kind invitation and the very generous introduction. So really my honor today to be here. I will share with you 2 stories. Okay. Can I see? Oh yeah, okay, good. So as Haibin mentioned, we have been working on SHP2 tyrosine phosphatase when I was a postdoctoral fellow in Tony Paulson's lab in Toronto. So I'm going to share with you some new data about SHP2 in liver regeneration and liver cancer. What is SHP2? You can see that here I listed a long name of companies. All these are big pharmaceutical companies such as Amgen and Novartis. Okay, here is one part of the list, and then second part, see, they're all working on SHP2 inhibitors. So what I can say is that you may not be able to find a big pharmaceutical because of 2 reasons. Number one, traditionally we isolate the inhibitors that target the catalytic domain of various tyrosine phosphatases. That turned out to be not a good choice because these inhibitors have low specificity. It can target many different tyrosine phosphatases. Number 2, these inhibitors couldn't cross the membrane. Okay, in 2016, I think Novartis made a breakthrough. They were able to isolate an inhibitor that can It's like it's basically allosteric inhibitor. It binds to the boundary between the SH2 and the phosphatase domain so that it locks the molecule in its inactive format conformation. Okay, it's not targeting the catalytic domain. That turned out to be very specific and it can easily get into the cell. So since that breakthrough, many companies are working on that. Another reason is, of course, SHP2 is the first identified oncogenic tyrosine phosphatase. So in the hope that you inhibit SHP2, you can block oncogenesis. But then now you wonder, what is SHP2? Why it's so interesting, right? I want to give you a very brief historic background. In 1970s, most of the cancer research labs were working on viruses that can cause tumor, the tumor viruses. All of these viruses are actually retroviruses, means what? They are RNA viruses, okay? And a very talented graduate student, Howard Temming at Caltech, worked on developing a quantitative assay to measure the transforming capacity of various viruses, okay? And he did a very good job in setting up the quantitative assay so that every lab later on used that method. But what I want to emphasize is that Howard Temming, was very talented technically. He's also very imaginative. Okay, in his PhD thesis, he proposed the theory how the RNA virus can replicate. He said that the RNA can go back to DNA, and then you follow the central dogma from DNA to RNA to protein. And then what happened is that the committee really liked his lab work. So you developed a very neat assay for everybody, but you are bullshitting about that theory. Okay, so because you did not have any evidence to support, and you are against the central dogma as a graduate student, that's too aggressive, right? But then you know what? He believed he's right. So right after PhD, he got an assistant professor position at University of Wisconsin Madison. And he got a small, tiny lab in the basement. He basically worked with a technician and proved that there is an enzyme can transcribe RNA into DNA. Now we all know that that's a reverse transcriptase, right? Because of that, he got Nobel Prize. So for postdoc graduate students, don't have to worry about any authorities. Propose your new theory, okay? Right, okay, so here, due to his quantitative assay and many other technologies, the field moved very quickly. And one great discovery made by many researchers was that they found you don't need the whole retrovirus. Only part of the genome is sufficient to cause tumor. And they call that part oncogene. So that's how we came up with the name oncogene, right? And of course, with so many different retroviruses, we found so many different oncogenes. And the most famous oncogenes are SUC and the RAS. Okay, so the next question, of course, why the retrovirus should carry gene that can cause tumor? Where are they from? That's a big question, but sometimes you only just need to do a very simple experiment. See, two young scientists at the UCSF, Mike Bishop and Harold Vollmer, did a Southern blot analysis. That's very simple, right? They used the VSUC as a probe to see whether it can light up or bind to something in the cellular genome. Yes, they found a band. Okay, and they call that as CSUC because it's in the cellular genome, right? With that Southern blot analysis, they got Nobel Prize. Okay, of course, then the next question is why SUC gene can transform the cell? What's the function of the gene product? Many researchers were asking that question, and Tony Hunter, that time, a young investigator at the Salk Institute, was able to demonstrate that SUC is a tyrosine kinase. So that opened a new field of tyrosine phosphorylation and the cell transformation. And so many people, including us, are still working in this field, tyrosine phosphorylation or dephosphorylation, right? But think about that time. Once you found SUC as a tyrosine kinase, what would be the next question? The next question is to see whether tyrosine phosphorylation is responsible for cell transformation, right? Okay, so a very talented postdoc, Tony Paulson, that time he was a postdoc in Steve Martin's lab at UC Berkeley. So he was able to identify a mutant of Fujinami sarcoma virus, a temperature-sensitive mutant. So he was able to demonstrate that as long as you turn off the tyrosine kinase activity, the whole virus won't be able to transform cell. So that was the first evidence that tyrosine phosphorylation is directly related to cell transformation. And here you can see for that Cell paper published in 1980, right? Steve Martin as a boss is the last author, and the Tony Paulson was the first author as a postdoc. And with that paper published, he was able to get a job, set up his own lab, and so that he can move his name from the first to the last. That's still the rule for postdoc graduate student. Remember that rule. Okay, so here you can see, that's a paper published from his lab. Tony Paulson was the last author, right? Ivan Sadowski was his very first graduate student. And also you can see from the title of this MCV paper, they continue to working on retroviruses that have transforming activities, right? And then what they did is they mutated so many different regions, chelated domain and the non-chelated domain. And they could do that because that time Tony Paulson's neighbor was Mike Smith. And Mike Smith was known for his discovery of site-directed mutagenesis, for which he got Nobel Prize, right? So that's why in science we say, who is your neighbor is very important, right? Okay, so they borrowed that technique from their neighbor, mutate the many different regions, and they were able to see that even if you mutate a non-chelated domain, that still can somehow affect or even abolish the transforming activities. So they call the chelated domain as the first domain, and then second one is a Src homology 2 domain, because that time, although they are working on so many different oncogenes, but Src was used as a prototype of oncogene. So that's why they named this as a Src homology 2 domain. So that's how we came up with the SH2 domain, right? So then when I joined the Paulson lab in Toronto, he moved already from Vancouver to Toronto. So I was thinking about the project, and I told Tony, say, if there are so many SH2 containing tyrosine kinases, there must be SH2 containing tyrosine phosphatase in balance, right? So how to look for that to test the hypothesis? I said, very simple. Use a primer in the SH2 domain, another primer in the phosphatase chelated domain. So in this case, if I do PCR and find something, it must be an SH2 containing tyrosine phosphatase, right? Very simple. We just test. If it worked, it worked. If it doesn't work, forget about it. Yes, very interesting. After 3 months, I was able to find a tyrosine phosphatase that contained 2 SH2 domains. So very interesting. That time, there were 12 postdocs in his lab. Everybody was working on the SH2 containing tyrosine kinase. In many cases, it only has 1 SH2 domain. So my molecule contained 2. So my existence was like more than justified in the lab, right? Okay. Okay, now I just also want to share with you how I enjoyed the science life that time. See, I joined the Paulson lab in September 1991, and 1 year later, we submitted a manuscript to Science, and it was reviewed, revised, accepted for publication within half a year. Okay, now here, that's a paper we did for Journal Club discussion. So it's published earlier this year. Can you see the original submission date, 2018? Took 5 years for resubmission, revision, and the publication. Think about if it's really new, then after 5 years, so the value, I don't know. And then also you can see how many authors there, right? And say that's how many authors here, right? So I don't know, we are all kind of frustrated, disappointed by the current situation, but nobody can do anything to change this disappointing situation, right? Okay, anyway, I say SHP2. is a tyrosine phosphatase, but it's an oncogenic tyrosine phosphatase. Means what? It promotes cell proliferation and the transformation. How it can do it? So here is a figure that we assembled for an invited review article that's coming out soon in Annual Review of Cancer Biology. So here, the mass equation is 1 minus what equal to 2. Obviously, the answer is a -1, right? Means what? SHP2 must inhibit and inhibit to promote cell transformation. And here are the possible targets. You can see that we listed quite a few. All of these SHP2 targets act as a negative regulator of the RTK RAS ERK signaling pathway. So you have a positive rule by removing a negative regulator. That's the idea. Okay. Now, here back to 20 years ago, we were trying to demonstrate that SHP2 is indeed a positive regulator of cell proliferation, try to find an in vivo evidence. So that's why we got into the liver research field. It's not because we were trying to work on liver diseases, but just trying to have liver regeneration as an experimental platform to test our idea. So here is the method that we used called partial hepatectomy. Right? In mice, you have 6 lobes. If you surgically remove the 3 big lobes, the 3 small ones will quickly grow out, and by 2 weeks, it will restore the original liver mass. Okay, then you can test in the knockout whether liver regeneration is accelerated or it's delayed, so that it will be a positive or a negative regulator of cell proliferation, right? And indeed, that's the most widely used experimental system. And using this system, many groups found these growth factors that are responsible for initiating liver regeneration, and also these transcription factors that are involved in the process. What's less known is the signaling molecule involved in this process. So a very talented graduate student, Emily Shapiro from France, she worked on this project, and we made the SHP2 knockout or its partner GAP1 knockout in hepatocyte and compared with the wild type. And you can see that for the wild type liver, our data match the literature, say hepatocyte proliferation reach a peak level at 40 hours after the surgery. And for the 2 mutants, you can see that they are almost identical because they're partnered, right? And also number 2 is that they follow the same kinetics for cell proliferation rate. And also number 3, see, in both knockout, the hepatocyte proliferation was reduced about 50%. So telling us this indeed is a positive regulator. So that's why if you delete it, you suppress cell proliferation. So that answered the first question we asked, but then there's a second question. Why some cells can still proliferate means what? Why deleting SHP2 did not abolish cell proliferation, right? And these dividing cells are also SHP2 negative cells. Why they can still struggle to divide? So that's the second question we address later on. So here, what we can see is that in the wild type liver, dividing hepatocytes are spread everywhere, distributed everywhere. But for dividing cell in the mutant livers, it's clustered. You either see almost no cell dividing in one area, and then in another area, these cells are all dividing in a colony or cluster. So that's why our original hypothesis is, okay, these dividing cells must be derived from a progenitor. It's expanding, dividing, so that's why they are in a colony. So with that hypothesis, we were looking for the progenitor cell markers. And one candidate is, of course, CD133, right? That has been used as a progenitor cell marker, and indeed, these colonies are positive for CD133 expression. So here, now what is CD133? It's a membrane protein with 5 transmembrane domains. It's a pentaspan plasma membrane protein, and it has been widely used as a biomarker for stem cell in various tissues. And also it's been viewed as a stem cell, cancer stem cell marker. because the expression is related to cancer recurrence and also is associated with poor prognosis of many cancer patients. Now, the problem is that SHP2 is also expressed in differentiated cell, and the SHP2 knockout mice had no defect in the stem cell progenitor cell compartment. Even it's viewed as a stem cell marker, then also more problem is that CD133 was also detected in cancer cells without any cancer stem cell properties. Okay, and the CD133 negative cells can also form spheres in vitro or tumors in vivo. These are the hallmark of so-called cancer stem cell. Okay, why that? But regardless of the controversy in the literature, what we can see is that it's now known, what is the function or mechanism of CD133 in either stem cell or cancer stem cell. So we are trying to address that question using our liver regeneration system because we found these dividing cells are CD133 positive, right? And here, okay, we were able to isolate the areas that are CD133 positive because they are dividing, compacting under under light microscope. You can even see a difference in these areas. So when we did a Western blot analysis, you can see that they are indeed CD133 positive, right? However, they are negative for EPCAM, AFP, SOX9. So there's no stem cell progenitor cell in the colonies. However, they are positive, very positive for HNF4 alpha. If you are working the liver field, you know that this is the hallmark of mature differentiated hepatocytes. Okay, now, here, the next question you may ask is, yes, whether you can detect the CD133 upregulation in other liver injury model. The answer is yes. When we injure the liver with carbon chloride, yes, we can do that. And also, the question, whether this upregulation can be detected in other cell types. The answer is also yes. We collected a number of cancer cell lines. As long as you inhibit the cell proliferation with SHP2 inhibitor or MEK inhibitor, yes, CD133 was dramatically upregulated. And also, I should mention that in the liver regeneration, right, we can see that in regenerating liver, CD133 was upregulated quickly, and when the liver regeneration is completed, it shut down. You couldn't detect CD133. So it's only transiently and rapidly induced during liver regeneration in the mutant, but not in the wild type. Okay. Now, here, another interesting thing, one postdoc, Kota Kaneko from Japan, he's very careful. He was trained very well at a university for his PhD program, about the cell type by immunostaining of different cell types in the liver. And he was able to see, say, hey, the CD133 positive staining, not only on the cell membrane, but makes bridges between the cell in these dividing hepatocytes and also in cell culture. And then on the highest resolution microscope, you can see the actual CD133 stained vesicles. And these vesicles are binding or associated with fibers positive for beta tubulin, right? And on the electron microscope or high resolution immunostaining, you can see that yes, indeed, we can detect the vesicles in the cytoplasm, in the juxtamembrane region, not on the plasma membrane. On the high resolution, on the low resolution, of course, you can see it on the membrane, right? And then when we isolate these vesicles, you can see that, okay, these are CD133 positive vesicles. They are negative for CD9, CD63, CD81. So these three proteins are hallmark for extracellular vesicles and exosomes. You can easily purify these vesicles from the cell supernatant. But for our vesicle, you can only lyse the cell and purify from cell lysate. So that's why together with the bridging between the cells, we give a name called intercell soul. It's not secreted into the supernatant, although sometimes we could detect small amount, but we couldn't rule out the possibility these are due to the cell damage, cell lysis, right? Okay. Now, here we isolated these vesicles and compared with the extracellular vesicle for RNA sequencing. You can clearly see the different pattern, right? Mainly, the difference is that for extracellular vesicles, they are enriched with microRNA. For our vesicle, they are enriched with messenger RNA. Okay, messenger RNA. And then here again, for all these staining, you can clearly see the bridge between the cells, right? And now here is the data for the single cell RNA sequencing of the regenerating liver, hepatocyte in regenerating liver. And then of course, because they are dividing, right? And if you look at the mitogenic messenger RNA, they are positive in every cell types. We selected these, right? And then in SHP2 negative cell, they are missing some messenger RNA. But if you look at these different SHP2 knockout cells, very interesting, they're missing different messenger RNA. One cell missing A, another cell missing B, the third cell missing C. And then if we look at the CD133 positive SHP2 knockout cell, then they are not missing anything. So that prompted us to propose a model, say, yes, because these cells are sufficient or are defective in mitogenic messenger RNA expression, yet they are strong to proliferate because after the surgery, liver is struggling to regenerate, right? The cells struggle to divide. We must divide. What can we do? So they share. If you are missing A, I'm missing B, then we share. We are not missing anything. I give you A, you give me B. So that's our model. Okay, and then here, let's compare the properties between exosome, extracellular vesicle with our intercellosome. One is positive for CD133, which has 5 transmembrane domain. The other contain these 4 transmembrane proteins called tetraspanines. So that's totally a different family of proteins, right? And I mentioned these vesicles contain microRNAs, and our vesicle contain mainly messenger RNA that are responsible for cell division, especially c-MYC, c-Jun, c-FOS. These are the primary messenger RNA that we detected. And one can be collected from cell supernatant, the other can only be purified from cell lysate. So that's why we believe these vesicles are involved in direct cell-cell communication. The exosomes, of course, are involved in long-distance communication, right? And one is induced by suppression of cell proliferation, yet the other one did not respond to that issue. And of course, that's like the most exciting story that we are working right now. And again, okay, no surprise, we had a very difficult time in getting it published. So that's why it's the first time that we deposited a manuscript to bioRxiv last year. And eventually the paper is published today. Okay, published today in eLife. Okay, I highly recommend eLife to you guys if you have a very interesting story and you don't want to be screwed up. Okay. Yeah, eLife. This time I can see that the editors are very good. Editors are prominent scientists. They know what's good, what's bad. Okay, they have the ability to judge, and then they send it to a reviewer. The 2 reviewers were all positive. And what's amazing is that reviewers also discussed with us the issues that there are so many questions to be answered, but they feel, yes, it's time to publish the first paper. So we will address these questions in the next 10 years. Not like we have to publish the first paper after 10 years, right? So that's the difference, okay? So now come back to SHP2 in liver tumorigenesis. So here I just show you one experiment. So using hydrodynamic tail vein injection of oncogenes. Usually we need to inject 2 oncogenes into the tail vein together with the Sleeping Beauty transposon. Okay, and the postdoc made a mistake. He told me, so unfortunately, I only injected 1 oncogene MYC, not like a RAS plus MYC. I made a mistake, but somehow I got a very interesting result. I see here, by injecting only one oncogene, you couldn't induce any tumor in the wild type liver, of course, that's expected. But in our SHP2 knockout, you can see there are so many tumors even after 4 weeks. I say, no, that's not a mistake. You made a big discovery, right? Okay, see, now we are very interested in this model, why one oncogene is sufficient to cause such a dramatic tumorigenic phenotype in the liver. So that time, another talented graduate student developed a single cell RNA sequencing method for the liver. So it's not that easy. For the liver, it's hard to dissociate. So once you dissociate the cell, you damage most of the cells. So it's not good enough for single cell analysis. She was able to do that by carefully trying different conditions. And then that's a Yan Liang, she was able to collect all the different, about 23 cell types in the liver. So then, I asked them to work together and did the single cell RNA sequencing by collecting the data from day 0 and the day 10 at the pre-cancer stage, and also 4 weeks. That time you saw the tumor, right? And then the interesting data came up. Yeah. Yeah, it's somehow it's frozen. Yeah.
PowerPoint is not responding.
Oh, okay, yeah. Oh, no. No, okay, it's
a PowerPoint and it seems been dead. Screen's great.
yeah, I
It
think
comes back.
Yeah.
Let's put it back on. We got to put it back on the web.
Oh, so also disconnected from the web.
Is it fire very big?
Yeah, I think so.
Doesn't want to drop out of it. About where were you?
Not there yet.
Yeah, okay, it's okay. It doesn't matter. Okay,
Then-
I can go back.
all right.
Yeah,
I'll start it from there.
Yeah.
Second.
Still no response.
Comma. Yeah, put you back on Zoom.
Share screen. The PowerPoint. Cancel.
Oh, it's still there. Yeah.
There we go.
Yeah, okay, good. Yeah, okay, great. Let's come back. So we did single cell RNA sequencing, and you can see that here we were able to identify 5 groups of hepatocyte, and we were able to determine this is a group of tumor cells, okay, because they are AFP positive, right? And then also they are HMYC positive. In this model, we injected the HMYC oncogene, so that's why these are tumor cells, right? What's interesting is that these cells are also SHP2 positive. So mention that this is in the SHP2 knockout liver. Okay, using albumin-free, so we deleted SHP2 in about 95% of the cells, or more than 95%, but still it's not 100%. So somehow tumors are selectively derived from SHP2 positive cells that escape the deletion by albumin-free. Okay, and that's confirmed by the Western blot analysis. You can see that in the SHP2 knockout liver, if you look at the normal liver tissue, they are SHP2 negative. But if you look at the tumor, they are positive for SHP2. So that's the most interesting data. And here, the reviewers are actually asked a very good question. Say, if MYC selectively transfect the MYC positive cell, then you got the result. But then that's not the case. We can see that at the beginning, luckily we did the single cell RNA sequencing at 3 different time points, right? At day 10, before the tumor development, you can see there's no correlation between MYC expression and the PTBN11 SHP2 expression. So meanwhile, MYC can get into SHP2 positive and negative cells, but only the SHP2 positive cells can be derived into tumors. So that's a very high selectivity, and it gives us a very good lesson. Here, now, when we transfect the mice with MYC plus the CMV-CRE, so the CMV-CRE will get into the same cell as MYC transfected, right? And then, SHP2 will be deleted by CMV-CRE if it escapes the albumin-CRE deletion. Then you can see that even in SHP2 knockout, there's no tumors. So what happened is that here, that's the biochemical data. We treat the cell with a SHP2 inhibitor, and you can see that MYC half-life was dramatically shortened compared to the wild type cell. So SHP2 is responsible for intact RAS-ERK signaling. That is responsible for MYC stability. Without SHP2, you don't have an intact RAS-ERK pathway, and the MYC will be degraded much faster. And here you can see the quantitative analysis, right? And then also, that was actually the original experiment we did. See here, we planned transfect the mice with MYC and the RAS. And you can see still there's more tumors in the wild type. In the wild type than the mutant, or in the mutant than the wild type, right? And then what's important is that when we transfect the mice with MYC plus RAS, then you got tumors that are SHP2 negative. So meaning what? As long as you have the mutant RAS together with MYC, you don't need SHP2, right? So that's a very clear evidence. And then now back to the original phenotype, you ask why there are more tumors. If you say SHP2 is required for tumorigenesis, why there are more tumors in the SHP2 knockout? Yes, that's an interesting question we answer. And here you can see that at the very beginning, when you have MYC transfected tumor initiating cell, okay, in the wild type mice, they are surrounded by F480 macrophage. So the tumor initiating cells were eventually eliminated in the wild type mice. That does not exist in the SHP2 knockout. Okay, so that's why here we clarify the controversy in the literature. So SHP2 is positive or negative. It's totally confusing why it can be both. Here, we are saying that SHP2 is required for cell intrinsic oncogenic signal. Without SHP2, MYC cannot transform the cell. But deleting SHP2 created a tumor promoting microenvironment. So that's why it's so complicated, and it brings us the issue. See, not only SHP2, there are quite a few other molecules that can either promote, it can be promoting and shutting down the tumorigenesis. Okay, and then, okay, let's see whether
This is shutting down. The computer is shutting down.
shutting down. Yeah, okay. Let's see.
Okay. It's maybe some software is updating in the background. It just shut down.
Great.
Do you have a computer?
Yeah, I have it, but the thing
We
that-
can't get it on the web.
Yeah, cannot.
I
get
guess
it.
at least this audience can. Battery is low. Oh, that's why. It's never plugged in. Okay. Figure out the reason.
It's not. Yeah, any questions? Like better first
So, hi, Professor. I'm a second year PhD student. So there's may not be a lot of things I don't know. So my question is, in your first half story, you shared like a cell share A and B, remember? And I just wonder, like, why does some cell have A and some cell have B? Like, why don't, if they receive the same treatment or not?
That's a signaling pathway, right? That's upstream of all the downstream genes. So somehow, we found that it's very interesting for the downstream genes. If you have a defective signaling from that pathway, that pathway is not completely shut down. Without SHP2, you still have the signaling, but it's not 100%. So when the signals are diminished, so somehow they are missing different genes downstream. That's what we found in the single cell RNA sequencing. Okay, and then, when they are clustered, so cells, have so many different mechanisms in stress response or in survival. For example, autophagy. Autophagy is a mechanism that cells facing nutrient defect, right? Not enough nutrients for survival. What can they do? They eat some of their own organs in order to survive. So of course, in that case, the cellular functions are diminished, but at least they can survive, go through the difficult period, and then do the next. So then for here, once you do the partial hepatectomy, we cut off 2/3 of the liver. So the 1/3 of the liver is not sufficient to supply the nutrients for the whole body. So liver must regenerate. In our body, there are only 2 organs that can regenerate. One is the skin. You cut the skin, it will be sealed, right? Let's say if you remove 1 kidney, can you have another kidney, or even cut half of the kidney? It won't regenerate another half. Only liver can regenerate, because without that regenerative capacity, we cannot survive, right? So it must regenerate, cells must divide, but there are just not enough material to divide. What can we do? Sharing. It's just, I feel like it's a similar mechanism as autophagy. It's basically a stress response mechanism. Okay, yeah, good. Let's hope that we can finish the talk by having no more problem, right? So here, okay, so SHP2 can be positive and negative. One is the cell intrinsic signaling, one is the microenvironment. So that's why I believe we thought out the whole problem. Then with that positive and negative rules, here, let's come back to all these inhibitors invested by the big farms, whether they're good or not. So in the review article that we just wrote this year, we just want to say, okay, here, yeah, okay, here, that's what we wanted to say. Definitely hoping for the best result. But if it does not work, let's say the SHP2 inhibitor is not as good as we expected in cancer therapy, can we blame the medicinal chemist? No. They are perfect inhibitors in terms of chemistry. They bind specifically to SHP2, they potently inhibit the SHP2 activity. It's just not good for cancer therapy because our thought, our idea in treating cancer is wrong. We are always trying to say, oh, wow, that's an oncogenic pathway, we target it, we block tumor. No, that's not the way to treat cancer. That's what I believe. So here, we can discuss more right later on. So now the second story is about liver cancer immunotherapy. Now here, I just want to put out a very provocative statement. Say, so far, liver cancer research has not contributed to liver cancer therapy. And I hope that I'm wrong, okay? Then, how about the current situation? Number one, in terms of small compound, sorafenib and all the related compound, these are all inhibitors of receptor tyrosine kinases. And we know that they are being widely used in the world around the globe, but they are not that good. Sorafenib, so the best drug in liver cancer therapy, it can only extend the patient's mean survival time for no more than 3 months. It's actually 2.7 months. That's the original data. When they expanded that clinical trial data, within the field, we know there's no difference between control and the treatment, between the placebo and the treatment. But that's a drug, very expensive, widely used in the world. Okay, then for immunotherapy with checkpoint inhibitors, we know that only 20% of the patients respond. Even within that 20%, most patients respond in terms of tumor size, but in terms of survival, also not that clear. So here, how we got into this field. So we were trying, of course, we are basic science researcher, right? So we were trying to understand the mechanism. See, using albumin crete to delete the target gene in hepatocyte, and then using MX1 crete to delete the gene in hepatocyte and the non-parenchymal cell. That's why many groups use. But when I look at this system, I thought that's not a fair comparison. Albumin crete is constitutively active in hepatocyte, MX1 need to be induced by injecting poly-IC, the synthetic double-strand RNA. I thought that's a big problem because I had experience in interferon signaling for my PhD work in Indiana University. You can see that I worked on interferon signaling. And actually, before joining the Paulson lab, I did 1 year postdoc with Brian Williams at the Sick Kids Hospital in Toronto. Also worked on the double-stranded RNA activated protein kinase called the PKR, the P68 kinase. So I believe when you inject the poly-IC into the mice, it just induces tons of interferon responsive genes, inflammatory cytokines. How can you do that? So that's why when we worked on this system, I was able to convince the student to inject poly-IC into the control mice, albumin-free mice, and the MX1 mice. And he showed that, see, as long as you inject poly-IC, you see tumor suppressive effect. It's not related to MX1 mediated gene deletion as reported in the literature, no matter where it's published, Cell, Nature, or Science. Okay, so that's why we focus on the tumor suppressing effect of poly-IC, and we found this reagent can induce and activate NK cells, dendritic cells, and M1 macrophage. So that's how we published our results. When you activate innate immune cell in the liver, it has potent tumor preventive effect. Here, you can see that we carefully use the word preventive inhibition. Why is that? Because it shows complicated results. See, if we inject poly-IC at the very early stage, it showed tumor inhibition. But then when we inject poly-IC at a later stage, let's say 5 months in the P10 knockout, you can see that it showed tumor promoting effect. It's very complicated. So that's why we conclude poly-IC induced MX1 is not an ideal system in liver cancer research or the liver research field, okay? And here we show that poly-IC can induce tons of genes in the liver, including PD-L1. By the way, I mentioned that PD-L1 is not effective in treating liver cancer patient because PD-L1 usually is expressed at a very low level in the liver. So with that induction, we tried a combination treatment. See here, induce the liver cancer by RAS and the MYC, 2 oncogene, right? And then treat the mice with either poly-IC or anti-PD-L1 or the combination. And then we see that only the combination show very significant tumor inhibition. Neither of the monotherapy did that effect. Okay, and some of the mice treated with the combination can survive very long for several hundred days without tumor. They are tumor-free survival. Okay, and when we look at the lymphocytes in these livers, we can see only after the combination treatment, you can see the CD8 T cells were dramatically boosted in the liver. So that's why it has so robust tumor suppressing effect, right? Okay, now here is our conclusion. In order to treat liver cancer, you got to simultaneously activate the innate and adaptive immunity. That's the direction that we will develop. Here, we did another simple experiment, inoculate the colon cancer cell into under skin or into the liver. So basically here, we are comparing the same tumor growing under the skin or in the liver and subject them to the same treatment, okay? Either poly-IC or anti-PD-L1 or the combination, right? And you can see why we do that experiment, I should say. This is a breakthrough paper done by Jim Allison's lab in 1996, right? They used the subcutaneous tumor models. And then here is a paper originally published by Dr. Hongzhou and the colleagues. Again, they use the subcutaneous tumor model. And if you look at the tumor immunotherapy literature, more than 90% of the studies are still being done in the subcutaneous tumor model. We believe there are defects, and this is an artificial tumor model. It's different from lung cancer, liver cancer, where the tumor grow out in the liver, right? So that's why we want to do this experiment. And here is the result. When the tumors are growing out on the skin, you can see that monotherapy, combination therapy, both are very effective compared to the control, right? And then if the same tumor growing out in the liver, then you can see only the combination work. Monotherapy did not have any difference compared to the control. Okay, so then we compare the subcutaneous tumor and the liver tumor for the different lymphocyte population. And here we look at the CD8 over CD4 cell ratio. You can see that on the skin, okay, on the skin, monotherapy, combination therapy shows same efficacy, but in the liver, only combination show a dramatic difference. Here and here, we are talking about CD8 over Treg cell ratio, right? So again, if we look at the immune memory cell, other more specific cell subpopulation, I don't want to go through every detail, but you can see clearly, only after the combination therapy, it shows a dramatic difference in the liver. That's quite different from the subcutaneous tumor, right? So that's why here, we make a conclusion in the liver, either for primary and metastasize the tumor, only if you simultaneously activate all these immune cell subtypes, you can have the good response. So the conclusion is that the response to immunotherapy or checkpoint inhibitor is dictated by the tumor microenvironment, not by the tumor. So that's how we conclude, and we believe, the direction is to look for the liver-specific immunosuppressive factor and remove these factors, we can achieve better therapeutic effect. And with that, I thank all the heroes in the lab, and thank you for your attention. So we'll be happy to take any question. Okay, thanks. Okay, yeah.
I have microphones for anyone who has a question.
Yeah.
Yeah, very nice talk. I have a question about intercell zone. So is intercell zone secreted? No.
Okay, yes and no. So we can detect a tiny amount in cell supernatant, right? But we couldn't rule out the possibility whether these are due to cell damage or due to secretion. No matter how, it's quite different from exosomes. For the same cell line, right? We can detect the exosomes, extracellular vesicle, by collecting the cell supernatant. But in these fractions, we rarely or barely detect the CD133 positive vesicle. And also another point is that for extracellular vesicle, they are positive for the tetraspanning with 4 transmembrane domains, right? So ours has 5 transmembrane domains. It's a totally different protein. And another important thing is that for these vesicles, they are dramatically upregulated in response to inhibition of proliferative signal. When the cells are suppressed and are struggling to divide, these vesicles were boosted, largely produced.
Okay, so how the intercell zone can regulate in the cell communication?
Yeah. So that's a big question we are asking. It must be involved in direct cell-cell communication, and then what is indeed the channel that can allow them to migrate between each other? We don't know yet, but here the first idea is that we detected these into the CD133 positive bridges between cells. Yeah, see here, clearly you can see that, right? See these are between cells. And they are, okay, another thing is that these bridges are positive for CD133, and they are also positive for an RNA binding protein. So these vesicles are enriched with messenger RNA, and we could easily detect RNA binding protein in these vesicles. So that's why what we believe that they are sharing. And also, of course, in the proposal, of course, it's still a theory at this stage, right? So we can see here, if you look at simply look at the SHP2 knockout cell, they are missing different messenger RNA. But if you look at these are the same liver, right? If you look at the CD133 positive SHP2 knockout cell, then they're not missing any of these messenger RNA. So that's why we are saying that they're sharing.
I see.
Okay.
Thank you.
Any last questions?
Yeah. From any question from the online? Okay, how can we see that? Okay, let's see.
I don't know. Maybe
Oh, okay.
there's no online question. Maybe I can ask.
Yeah. Okay, good.
So, Gensheng, when you said this, when you knock out SHP2, it may have anti-tumor activity in the tumor and also may promoting tumor promoting environment. When you say that, are you suggesting the SHP2 within the liver's hepatocytes can do that, or SHP2 in, let's say, immune cells are promoting the tumor forming environment?
Okay, that is a great question. So here, I just want to say that, with genetic model, we can clearly define the biological function of SHP2 in each cell types, but then that may not be able to explain pharmaceutical outcome. Because when you inject the SHP2 inhibitor into the mice, it will work on the tumor cells and also immune cell. And remember, SHP2 is expressed in every cell type, right? And there are publications that SHP2 bind to PD-1, regulate the negative pathway in hepatocytes. So that's why here in the review article, we can only be cautiously saying that we don't know the outcome yet. We are hoping for the best. We are preparing for the worst, right? So that's the idea, I guess. You don't know how it works, right?
All right. Any more questions? All right. Thank you, everyone. Thank you, Gensheng, for the great talk.